Swayam Prabha. Digital India. Educated India.
With our agenda today. So remember that I started to talk about CRISPR with the idea of having a way of counter-selecting in recombination. Because, as I said, one thing is to distinguish a mutant from a wild type on the basis of a phenotype. That's very simple. I mean, if the phenotype is that you grow on this or not grow on that, then that's very simple. But if you don't have a visible way to monitor and to visualize the differences between the wild type system and the mutant, then how do you do it? Okay, so the good thing about CRISPR is that enables you to distinguish between a mutation and a wild type on the only basis of the sequence, okay? Nothing else. You don't need to have any phenotype to bring about that change. Okay, so let me see here. Okay, well, I think that. Okay, so in practical terms, what you have is a deletion protocol that is simpler because it requires recombinant oligo for just one. Then select the spacers, design oligos, clone them in different plasmids, transform your strain, grow the cells, and confirm the deletion. So all this takes just a few days rather than few weeks, as I said before. Well, there are some details on some of the plasmids that people have been developing to use CRISPR precisely for this type of applications. You have one of them, for instance, where you have Cas9 being expressed together with the track RNA and with some places where you can clone different segments for production of the RNA that will interact with this other RNA with Cas9, and then it will act as a nuclease. And some variants of it where you can distinguish, I mean, you can, for practical terms, you can have the two separated. I mean, if you go through a literature, there's a tremendous variety of systems based on CRISPR-Cas9 that eventually they do exactly the same. So in our hands, and for handling Pseudomonas puti, that is our favorite bug, we prefer to use this two plasmid system where we have, on the one hand, one plasmid that expresses the tracing DNA and Cas9 in a constitutive fashion, and then we have this other where we engineer the expression of the target DNA, the target RNA here. Okay, so and they are compatible because they have different origins of replication, and they normally work very well. So for a given application, what you do is that you use these two plasmids in the laboratory, then you just co-transform simultaneously the mutagenic oligo with the plasmid that produces the tracing DNA and the plasmid that expresses Cas9. And in that way, what happens is that when everything gets together, the only way that plasmid, that cells can survive is if you have executed the mutation that is carried by the oligo here, okay? So that's something that is very nice. Okay, so if you go through a classical deletion protocol, then as I said before, it will take 2 to 3 weeks in the best case scenario. If you go through recombination based CRISPR deletion or CRISPR some other, say, editing, it will go to 1 week, okay? So that's something that, you know, in terms of saving time, is a considerable saving. There's another screw term that I want to mention because I think it's super interesting. That is what has been called precision genome editing, okay? So precision genome editing means how can you, with a minimal manipulation, a minimal preparation of things, how can you really enter very, very specific mutations in a good part of, in a given location of the genome. This is something that, of course, has been applied in bacterial cells, but also this is the very same concept that has been applied for gene editing in mammalian cells, in animals, and in other types of complex biological systems. And this is possible because of some properties and some, say, new proteins that have been discovered recently that can be engineered to do something very nice. That is the following. We have just explained to some extent that in the CRISPR system you have the Cas9 protein. The Cas9 protein recognizes a specific site in the genome because you have a guide RNA and you have a nucleolytic activity that will cleave the DNA there. However, it is possible, and it has been made, to have mutants in the nuclease domain of Cas9 that maintains the ability of this whole protein to bind to a given site of the genome, but without cleaving, okay? So that means that now we have a big protein, this Cas9 protein, together with the RNA, that together will go to a location in the DNA and will remain there. And it doesn't have a nuclease activity. Because of that, it's called a decas system, decas, okay? So it was, before it was a nuclease, but now it is not a nuclease. On the other hand, in the last few years, a large number of proteins have been isolated that are cytosine deaminases. Okay? Cytosine deaminases are enzymes that are present in many cells that will find cytidines in the DNA and will deaminate them to convert them in uridine. And then this uridine, upon subsequent cycles of replication, will be converted or will be recognized as a thymidine. Okay? So a cytosine deaminase is able to convert cytidines eventually into thymidines. Okay? And these are proteins that, as I said, have been discovered in various contexts and so on. Now, what if, as we are smart, you make a protein fusion between Cas9, the variant of Cas9 that is unable to cleave DNA, and a cytosine deaminase? So what happens is that now you have a complex protein that on the one hand is able to recognize a specific sequence in DNA, but on the other hand, in the proximity of web location where the complex goes, the bases that are, well, the cytidines that are accessible to the cytidine deaminase will be converted into T's, okay? So C will go an U, and then you will have a specific change there. And this will be very specific because the complex, the geometry of the complex of this hybrid with the DNA will be very well maintained, and therefore the possibilities of the cytosine deaminase to execute its activity on the cytidines will be limited to the window that the architecture of the complex enables, okay? So let's take a closer look to this. So this is a typical fusion between Cas9, decas9, and then a cytosine deaminase, okay? So first of all, well, you may have a linker between the two, longer or shorter, and that will determine the window of action of the cytosine deaminase. You have a long cytosine deaminase, a long linker between one protein and the other, then the window of activity can be relatively long. But if you have a limited size in the linker, then it's like, you know, you have the protein sitting there, and then the cytosine deaminase will be able to act only on the cytidines that are found adjacent to the place that you are binding. So, so this is the situation, and also there's an additional trick, and is that in this scenario, in many cases, you add one more domain to the protein hybrid that is an inhibitor of an enzyme that is called uridine glycosylase, that what it makes is to repair the changes from C to U. So just to avoid that the changes are repaired, the protein engineers have added to the complex this particular addition there, okay? So then this is a kind of great machine because you can take the complex and target the complex to a given location of the chromosome, and then without any recombination, without anything else, then chemically or biochemically, a change between C and U's will happen, and that will be the result of the whole operation. Okay, so that's something that is very, very nice. So there are base editors. Not only are cytosine deaminase, there are other base editors that are adenine deaminase. But by the way, the complex between Cas9, dCas9, and cytidine deaminase or other deaminases is what is called a base editor, because the base editor suffices by itself to enter mutations at a given locations of the genome just by themselves, without recombination and with anything. Okay, so when you have CRISPR-Cas9, the complex will cleave the DNA, but when you have this kind of variant where you fuse, or with a dCas is fused to a cytosine deaminase, then you can enter specific mutations in the genome without any intervention of any other mechanism. Obviously, in bacteria, well, this is perhaps not so, say, spectacular, because we have many other ways, as we discussed before, to enter different types of mutations in the genome. But for instance, in eukaryotic cells, this is a kind of magic trick, because you can go into the genome, and by doing this trick, to enter and to revert mutations that are known, for instance, to cause diseases. And therefore, this is one of the most promising, say, approaches to gene editing in animals. And well, you may recall this story years ago of this Chinese researcher called He that created two babies by manipulating the embryos and using exactly this technology to enter in some specific genes mutations that make them immune or completely blind to infection by HIV. Okay? And well, the technology obviously is not as simple as I'm telling you, but this is the ultimate basis to use CRISPR-Cas9 technology and the Cas9 technology together with a guide RNA and these base editors to edit the genome, the specific gene of even a human being. There are various issues there. I will not get into that. There are, ethical issues and risk issues and everything, because one of the main problems when you deal not with bacteria but with animals and so on, let alone humans, is that you are not secure, you are not sure about off-site effects. Okay, so you think that when you have that situation, the only target DNA that will be edited is the one that corresponds to the complex that you have engineered, but it may happen that this complex will go somewhere else and will create also mutations that you don't expect. Okay, so these are things that require some further discussion and so on, and it's not trivial that we can use this technology for gene editing, for a kind of preemptive gene editing. A different thing is when you have already the disease and the disease is serious, and then you have very few therapeutical choices, and then the only choice might be that you make a kind of gene therapy and change the structure of the protein that is giving you a hard time by changing the structure of the gene. Okay. Well, but this is what happens in eukaryotic cells and in mammalian and so on. But in bacteria, you have an interesting application, and is the possibility of entering many mutations at the same time in a given metabolic engineering project. Okay. So, for instance, in this case, and this is very recent work. It came out just a few months ago. So what these researchers did was to target many different mutants here that they wanted to have for having a variant of Pseudomonas putida that would have specific catalytic properties. By the way, every time I mention Pseudomonas putida because it's the bacterium that we are more familiar with, but just remember that every single technology that I'm sharing with you today is perfectly applicable to basically all types of bacteria. Okay? It's just that to give you a more realistic example, I use the ones that I am more familiar with. So in this particular case, what these people did was to trying to get simultaneously 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 mutations at the same time. And the mutations that they wanted were null mutations, namely to kill the genes. How do you kill a gene? By entering a short mutation, by entering a stop codon in the gene, okay? So you have the sequence of the gene, and in the middle of the gene you put a stop codon. That means that when the gene is transcribed and expressed, then it will not happen, it will not be expressed. So what these people did was to make this multiplexing sequence of intervening DNA sequences that were corresponded to guide RNA that were connected by these repeated sequences. This is produced as a long RNA that is processed in vivo to produce smaller guide sequences. The smaller guide RNAs make a complex with Cas9, and then the same, and each of them results in a base editor that will enter, in each case, stop codons in each of the genes, okay? So that's a wonderful tool to enter many null mutations in a given bacterium, in many cases for the sake of metabolic engineering. In this particular case, it was for making Swayam able to deliver a reaction of biotechnological interest, okay? So that's something that is quite nice. Okay, so let's recap a little bit things that we have seen today and in previous days of genetic operations that you can do with plasmids, okay? So plasmids, you can have many types. Remember the SIVA collection that we discussed yesterday. You can use them for cloning, you can use them for promoter probing, for expression, and as we have been discussing today, you can use plasmids for the most part also for as editing modules and editing tools, whether they are based on SC1 based homologous recombination, whether we use double-stranded DNA recombination, or whether we use CRISPR-Cas9 based recombination for different types of metabolic edits, you know. So this is something that is quite handy, and you see that plasmids basically are the workhorses for nearly every type of, say, operation that you make in for genome editing in bacteria and in many other types of, say, genetic engineering endeavors. Okay, so I'm going to spend the last part of the discussion today revisiting the issue of transposons as tools for engineering genomes. As you remember yesterday, transposons are a way to enter new genes into the chromosome of different bacteria because of their abilities. And I just mentioned on passing, or well, not on passing, we discussed yesterday in great detail the TM5 and some of the things that you can do with TM5, but that's not the end of the story. Okay, so I want in this last half an hour of the class today to revisit some of the many utilities, applications that you can have with mini transposons for genome engineering as well. Okay, well, what are transposons? We saw that on pacing before. They were discovered a long time ago because they generate phenotypic and genetic diversity. We will not get into that. And well, as we also mentioned on passing, DNA transposons or DNA sequences that can move around. We have an issue here. So, hello? 10 minutes, no? 10, okay. So, yeah, but you have eliminated the window, but not the problem, right?
No, it's not.
Oh, not a problem? Okay, hope so. Okay, so transposon, a sequence that can move, etc., and it's because of the action of a transposase. Okay, the one that we discussed yesterday was the TM5 transposase, but there are many other types of transposition and transposases. Some transpositions are the result of what is called a conservative cut and paste. In other cases, they are replicative. Okay, so that means that sometimes you have the donor replicon, and then the transposon goes to the target, and then the earlier transposon remains where it was before, and then it gets a copy of it. But in other cases, you have a donor and a target, and then the donor eliminates the transposon once the transposon goes somewhere else. So you have a complete variety of transposons for different applications. The ones we saw yesterday was this TM5. TM5, I will not get into that. We have all the genes and everything. I explained you how you can go from here to a mini transposon. But there are two other transposons that are quite, say, handy in synthetic biology and in general in genetic engineering of bacteria. One of them is the so-called TN7 that originally delivers resistance to tetracycline. The structure is very similar to the TM5, so you have also insertion sequences. In this case, insertion sequence 10, no insertion sequence 15. And then, as was the case also with the TM5, the insertion sequence to the left happens to have a transposase that is inactive, okay? Whereas the one to the right is active. So this is a structure that you can find in many naturally occurring transposons. You have something to the left, something to the right, and then something in between. The difference here is that you have other antibiotic resistances. We have also, and I will not tell you the details, made mini TM5 transposons exactly with the same philosophy, with the same basis that you have made TM5 mini transposons. And you can put inside the TM5, the mini TN10 transposons, all types of segments of DNA that will work also very well for different purposes. But one transposon that is quite good for diverse applications is TN7. So TN7 has the property, I mean, you have also some antibiotic resistances, and you have here a complex transposase that is not just one gene, but it has a bunch of different genes that assemble the complex that is necessary for transposition. But the advantage of TN7, or disadvantage, if you want to take it, different from TN5 or TN10, is that TN7 will target just one specific site in the genome, okay? So TN5 will jump all over the place, but TN10 will jump all over the place, but TN7 will go to specific locations, okay? And remember that you can make mini transposons out of them. Okay, so I'm going to show you a couple of applications where you can really see the power of mini transposons for genomic engineering, okay? So one of them is based in one of the plasmids that I mentioned yesterday, that it was PBAM1 that has a mini transposon that is compatible with the SEBA platform, okay? So this transposon, that is the best optimized transposon that we have produced so far. What we have done is to remove non-functional sequences. We have improved expression. We have minimized the occurrence of restriction sites. We have bordered the modules with rare restriction sites, as it was the case with the SEBA vectors. And then we have added a multiple cloning site with 15 restriction sites. So basically, this is a very good transposon vector, because basically you can add all types of heterologous DNA sequences to the cloning sites, and that sequences will be eventually transposed to the target strains. And we'll have made also some improvements at the borders of the transposons to increase the transposition. In the case of the TN7, then we also have different variants of TN7 that for some applications are better than TN5. So they are orientation specific. They go to specific locations in the chromosome of the target bacteria, to a place that is called at TN7, so you go straight into that place. The frequency is very high, stables are very stable, they are very wide, broad range, many gram-negatives, different pseudomonas, whatever. And the TN7 transposase can also act in trans, as in the case of TN5 and TN10. And what is very interesting is that because the insertion goes always to the same place in the genome of the target bacterium, then you can make a collection of isogenic strains that are identical excepting for the nature of the insertion that is precisely in the T7 site, okay? TN7 site. So that's something that for some applications is very, very useful. So in TN7, there are many types of delivery plasmids. As you just heard, the advantage is that in the case of TN7, the transposase acts in trans. So remember that in TN5 and TN10, the transposase acts in cis. So the transposase has to be provided to the cells in the same construct where the transposon is. In the case of TN7, you can split physically the plasmid that has the transposon and the plasmid that has the transposase, okay? And that's very good because you can really do things that will be difficult with TN5. So there's a whole bunch, a whole collection of TN5 variants, and the advantage of the TN7 is that you can combine these variants in such a way that they are perfectly isogenic, okay? So that's very good. Okay, so let's take a look to what are the applications of mini transposons for different endeavors in bacterial genome engineering. Well, number one, you have random mutagenesis. This is the most trivial one. This is not really genome engineering. It's just another application of transposons. But what is more important is that you can use transposons for integration of heterologous DNA into the bacterial genome, whether through random integration in the case of TN5 and or a specific integration in the case of TN7, okay? So in the case of random mutagenesis, well, it's very trivial, and we commented that on passing yesterday. Then you can use the technology for various purposes to study gene functions or to correlate a mutant phenotype to a specific gene. We have various things, but mini transposons, you know, we discussed that yesterday. I will not get into more detail now. The disadvantage is that mini transposons interrupt genes and therefore can alter the expression of surrounding genes under what is called polar effects. And that means that if you want to have a predictable outcome of your genetic engineering or genomic engineering project, then that might not be the best way to go. So for random integration, you have the mini TN5 and the Mariner system, or the TN10 and the TN10 as well. So you have some choices that you can make. The differences have to do with the frequencies of inserting different types of DNA sequences. TN5 has the tendency to go more into 80-rich regions. Mariner is more promiscuous. It can go to different locations. And in the case of TN7, it's not possible to go to different locations. It will always go to the at site TN7 that is in the genome of your target bacterium. Okay, so you can kind of make a list of possible examples, possible situations where mini transposons can really be useful, and I will show you just some of them. So, for instance, in this case, what we had is a situation where we wanted to identify genes responsible for a production of a pigment, okay? So in this case, the phenotype is very clear. You make the transposition, and then at the end you plate on a selective media and observe the phenotype, okay? So I don't know if you can see it there, probably not much. And in the mating, you have a recipient strain, you have a donor strain, and you have a helper strain for formation of these three-parted matings that we discussed the other day, okay? So as the outcome of the mutagenesis is a visual thing, because the gene that we wanted to target with the genome, with the transposon, was involved in the production of the color, then all you have to do to see whether the insertion has gone to the gene that you want is to take a look to the plate, and even though the quality of the picture is not very good, you see that the background here, most of the colonies are bluish, and once in a while we have some colonies that are white, okay? So that's an indication that the transposon has hit one of the genes involved in the production of that particular color. Okay, one important, say, consideration in this, when you make this type of mutagenesis is how many mutants do I need for generating a good library, okay? So this is just a purely mathematical calculation. So in a bacterium you have a number of genes, maybe 500 in the case of smaller genomes, or 5,000, even 10,000 in the case of larger genomes. So given that a transposon will hit randomly in all possible locations in the genome, how many insertions do I need to have a representation of insertions in every location of the genome, okay? So you can make a calculation, and you have to, I mean, it's not true that you have 5,000 genes, you need 5,000 insertions, right? You need 10 times more to ensure that the whole gene complement, the whole genomic complement has been covered with these things. Okay, so obviously the more mutants that you have, the more insertions that you have, the more mutant that you get. In the case of Piputida, for instance, you can make a calculation, and that goes that if you enter randomly 10,000 insertions in the chromosome, chances are that the coverage of insertions will be 47%. If you go 30,000 insertions, you will cover 85. If you have 50,000 mutations, you will have 96, and you need, at least 100,000 mutations to cover practically the whole genome. Okay, this is a pure mathematical calculation, and it does not really, is something indicative. Okay. Okay, so, but as I said, one of the main applications of these transposons is to insert new segments of DNA in the genome of a pre-existing chromosome, right? So, for instance, in this case, there's a project that we started time ago to take genes for production of polyhydroxybutyrate. That is a plastic, a bioplastic, that is produced by this bacterial species called Cupriavidus necator, and then produce an E. coli strain able to do that at high levels, okay? And to that you need 3 genes here, A, B1, and C, A, B1, and C. So what we did was to take a fragment of the chromosome of Cupriavidus necator and insert this segment into the cloning site of a transposon vector, okay? So this is one of the standardized transposon vectors with the cloning sites, and then you take this fragment, you put it in the cloning site, and at the end you generate a mini transposon where you have the outer site of the transposon with the sequences that are recognized by the transposase, the downstream site similarly the same, and then in between you have the 3 genes of interest together with a selection marker that in this case is gentamicin here. Then what you do is that you take this segment or this construct, you put it in, you make it transpose into a target bacterium, that is E. coli, BW, whatever, with that carries one gene that would interfere with the formation of bioplastics, and then now you select or screen for clones with a desired level of expression, okay? So this will be one of the most, common, cases where you use transposons for genome engineering, because you have a segment of DNA that you want to introduce in an stable fashion in the genome of a target bacterium such as E. coli, okay? And then you make the transposition, and then you select the clones for a desired level of expression. Okay, what happens, and this is something quite intriguing that might require some explanation, is that you have many insertions in the chromosome, you can have thousands of them, but not every insertion will deliver the same level of the product that is encoded by the pathway that you enter in the chromosome here, okay? So some of them will be highly expressed, others will not be expressed at all, and others will be very poorly expressed, okay? So in this case, you have a situation where the insertion of the transposon will give you a kind of landscape of expression of the phenotype that you want to enter into your construct here. But there are other cases where you don't want to have this diversity of expression at different levels because of the different locations of the transposon in the genome. What you may want to have is everything located exactly in the same place. And in that case, what you need is a transposon that goes only to one location of the chromosome, and that transposon is TN7. One of the most typical, applications of TN7 for this type of constructs is to have variants of the same strain that are tagged with different colors. Okay, so that's something that is become very popular because the ATT TN7 site where the T7 transposon goes happens to be in a place in the chromosome that is completely neutral, and therefore you can insert there heterologous DNA segments and still the cells will be viable and will not even notice that you have introduced there a segment of your choice. Okay, so this possibility of having isogenic strains that differ only in the nature of the DNA that is in the TN7 site enables having a collection of bacteria that are identical excepting for something a bit different that you introduce here. So this is the most favorite, say, approach to have bacteria tagged with fluorescent proteins of different colors. And this is something that has been done numerous times, not only in my laboratory, but in others' laboratories as well, in which you just use a TN7 together with a GFP or variants of the GFP to have all types of color bacteria. So, and when you have bacteria of different colors, then you can follow them when you release them in the environment. You can see whether they colonize the plants or they survive in soil or they transfer their DNA. I mean, there are many, many different possibilities that you can pursue once you have a bacteria that you can track or trace with a given color. So, in fact, one of the things that are more interesting is to see the dynamics of the interaction of bacteria when they touch with themselves or when they form clusters or wherever, because they are tagged with different fluorescent markers, and see how they interact and whether they form structures. So this is something that becomes very, very useful. Okay, so let's make a little summary of things that we have been discussing today. I insist that I know that today has been a very heavy, loaded talk, not precisely the most exciting, but again, I insist that you need to know the tools, not only the applications where very flamboyant applications. It's important that you know the mechanism of the tools that you use for genomic engineering, because this is the tools and the technologies that enable you at the end to program cells for various purposes. Okay, so as professional synthetic biologists, we have to concentrate and to consider not only the final things. One can have beautiful ideas, or one can be done at the end of a given project, but the basic foundation and what enables things to happen is to have a complete control of the technologies and the tools that you have on top of your table. You have a good tool, there are many things that you can do. If you know how to use your tools, there are many things that you can do. If you don't know how to do them, you can have beautiful ideas, but they will never happen. Okay, so recapping a little bit, the various lectures that you have heard about genetic tools, some of them more traditional, some of them less traditional. Then, first of all, you have a bunch of plasmids, a bunch of vectors to clone genes or operons, and this is the basic operation in genetic engineering, to clone genes or to clone clusters of genes and to have them under your control. Then some of these plasmids, some of the genetic tools, mostly plasmid-based, are useful for studying transcription, because we have a large number of plasmids and vectors that are promoter probes, in which we have a promoterless reporter that can be inserted upstream with a segment of DNA, and therefore the readout of the reporter will be an indicator of the activity of the promoter. Okay, so this is something that obviously is also in our agenda. Then we have another type of plasmids that are adequate for heterologous expression of DNA in bacterial cells and in other biological systems, because we repurpose the regulation that we observe in some naturally occurring regulatory nodes of bacteria. We take it to the laboratory and we streamline the different components, the different parts of that expression node, and then we use that for heterologous expression. A heterologous expression is a part of cloning the genes. Heterologous expression is the main tool that genetic engineers and synthetic biologists have for making different types of constructs. And then we have a large number of tools for genome editing. So the most basic of them is the one based on homologous recombination that depends on the so-called REC-A based system of bacteria. That is a system that naturally is used for fixing damage to DNA, but it is also useful for, in a different context, for using, for swapping segments of DNA between a plasmid donor and a genome recipient, and therefore it enables a large number of manipulations. With the disadvantage that if you go to this type of homologous recombination based on REC-A, the system works very well, but is very slow. It will take a lot of time to produce different mutants and so on. You can also make mutations by making mutant libraries, but it enables also homologous recombination to insert heterologous DNA at specific locations, and also use transposons at random places. Okay, and different technologies enables us, either with plasmids or with recombination, to insert DNA at specific sites and edit specific sequences, mostly with the tools of this precision engineering that I told you with CRISPR-Cas9. Okay, so that's something that is quite good. Okay, so I know that we will not have more sessions for presentations of papers, so that's fine. Today will be the last session, we will have different presentations. And, but however, I want to mention some papers that are connected to the presentation that we had today, and that I believe are important, or is important, for documenting, if you are interested, many of the technologies that we have been discussing today. Okay, so we have 4 papers here, and the 4 papers are in the web page and the repository of papers that we have in the Dropbox that I have habilitated for this course. So one of them is this one here, TM-based device for calibrated heterologous gene expression in Pseudomonas putida. So this paper is about the use of TN7, that, if you remember what I mentioned before, is a transposon that enables to place insertions of DNA always in the same location of the genome of your target bacterium. So in this case, what we made, and Sebastian and others made, was to make a collection of promoters. And then each promoter was located in the same place of the genome of Pseudomonas putida because of TN7, together with a reporter gene. So by doing that, we have exactly the same context in all constructs, excepting for the nature and the sequence of the promoter. And therefore, that enabled to calibrate very well the activity of the promoters, because we could really compare all the conditions regardless of the context, because the context was always the same in every case. So that's something that you may get some inspiration in case that you are curious to take a look to that paper. Okay, then there's this other paper that came out just a few weeks ago, and it's super interesting. And it's this idea of using multiple mutants, of creating multiple mutants in the genome of Pseudomonas putida, but could be applicable to many other bacteria, with this cytidine-based editing. So in this case, what the authors do, and I showed you before some slides that come from their paper, is to enter 10 different stop codons in the structural genes of 10 different enzymes that they want to kill for having a strain that is useful for metabolic engineering. Okay, so the kind of nice aspect of this work is that they enter simultaneously 10 mutations, and they all seem to work very well. Okay, then finally, I want just to mention that there are these two papers that is interesting to read. One of them is to use a CRISPR-Cas9 base, CRISPR-Cas9 for counter selection of mutations that are entered in the genome through recombination. So in this case, the thing is that the mutations that are entered in the genome do not give any visible phenotype. But by doing this trick of CRISPR-Cas9 based counter selection, then the authors can distinguish between the wild type sequences and the mutated sequences. And therefore, that simplifies tremendously the screening of those mutants that are expected to be produced through this technology. And finally, I would like to mention here this paper that is there, I think 2009, with this system that is called MAGE. You know, remember MAGE, Make America Great Again. So I think that at least the acronym you will not forget, and that multiple genome engineering and multiplex genome engineering accelerated evolution is the platform that enables to enter again many different mutations by transforming into growing cells mutagenic oligos together with expression of a recombinase that will facilitate the entry of these mutagenic oligos in the replication forks of the chromosome, and therefore will enable as well the inheritance of different mutations at the same time. Okay, so these are things that we that in all cases are very, very useful. Okay, well, I will return to this because then we have to think who has talked, who has not talked, and how to proceed later. I will return that later. But anyway, so I think that we are now at the end of this of the formal lecture for today. And I insist that I know that today it was too heavy and so on, but but you really have to know the tool that you have in your hands, because otherwise there's no way. I mean, you cannot make a nice painting. You have the right paint, the right brush, the right concept, the right these things, and this kind of nitty-gritty of genetic engineering and synthetic biology is precisely the type of tools that I have explained to you today. One problem that occurs with the tools is that tools are changing all the time. Okay, so when homologous recombination was proposed as a method for genomic engineering, it was a super big thing, So, and no one thought that there were many downsides. And then when the first, say, version of recombinering was put forward, wow, that people believe that it was the ultimate technology to make changes in the chromosome of bacteria. But then when single-stranded DNA recombinering appeared, wow, everyone was absolutely fascinated that you could do that. And then when you would combine that with CRISPR-Cas counter selection, wow, that it will be the ultimate way to do things. And now, we are, many people say that everything I've told you is completely useless because then will be genome synthesis, So why you want to have systems for genome editing when you can write things up front, right? So I don't know what you think about it, or maybe you don't have an opinion about it, but the thing is that sometimes the technologies grow so fast that there's no time to really nurture and capitalize and leverage entirely the technologies for so many things that could be done, right? So you have and we face that in our laboratories all the time, because our laboratories have specific problems. We want to produce in the laboratory a bacterium able to produce this compound or to degrade this compound, and at the end that theoretically you can abstract the problem, and that to get a strain that produces this compound you have to have 10 different mutants in the chromosome, you have to enter 5 different genes, and you have to enter 20 different promoters, and I mean exactly what you have to do. But how you do it, the technology on how to do it may change completely over the years. And therefore, sometimes you start a project with a technology, and then in the middle of it, a much better technology appears, but you have already invested the time in that earlier technology. So what do you do, right? So one example that I have experienced, and is a kind of, I wouldn't say a sad story, it's a kind of a story that one learns during the experience, is the transition that happened years ago between DNA chips and transcriptomics based on deep sequencing, okay? So when DNA chips were first proposed, wow, it was like the final ultimate response to monitor transcriptional profiles of cells simultaneously, right? So you had the chips, you had to be printed, and so on, and the machine was beautiful. Everyone bought a DNA spotter, and laboratories went crazy for this type of technology. But a few years later, it was just deep transcriptomics and ultra-fast new generation sequencing, and that was the end of the DNA chips, And then an entire technology that supposed a lot of investments and these things went completely down the drain, right? So this is something, well, this is not, what I showed today is not so dramatic as with other technologies, but the thing is that we have to live with the fact that sometimes we start a project with a technology, and then by the time we want to publish that thing, then the technology is completely obsolete, right? And that happens all the time. So there's one specific project that I'm facing these days, exactly the issue, and it goes like this. So at some point we were interested in understanding the basis of this of why Pseudomonas putida is so sensitive to desiccation. So some bacteria, I mentioned that on passing the other day, can resist desiccation. Desiccation is lack of water very well, and they remain viable, but other bacteria are super sensitive to desiccation because they trigger oxidative stress response. They produce a lot of reactive oxygen species, and then cells are dead. And Pseudomonas putida is very sensitive to desiccation. So we wanted to understand what happened. So what we did was to use chips. By that time, we're talking about 10 years ago. And then one sabbatical visitor came to my laboratory. She spent a lot of time using this with chips and everything, and we had wonderful transcriptional profiles of genes going up and down and everything with chips. But then what happened is that this lady, she was a lady, left the laboratory, and then she forgot about the project, and the project was in the drawer for 2 years or 3 years. And then when we wanted to continue the project, then the technology had changed completely. So the chips were a thing of the past, and the only acceptable results that journals would take would be data from deep transcriptomics. And okay, so what do you do with that, right? I mean, all the data that you have with other technologies, what do you do with that? So that's something that happens all the time. And also for mutations, until 10 years ago or so, making mutants with double recombination was the standard, okay? Now, if you send a paper saying that I have created these mutants through double recombination, people will think that you are, that you are a Neanderthal, that you are not updated on the most modern technologies. So there's this issue of, technological advance. In some cases, is a question of fashion, So because the final result is the same, you will have the mutant, but it's just that if you do it through a traditional procedure, you will be considered to be not sufficiently updated, with the latest fashion in so on. And many of these technologies are also subject to fashion, So these days, if you don't use CRISPR-Cas in your projects, you are nothing, I don't know what you guys are doing, but please make sure that anything you do incorporates CRISPR-Cas, because if not, forget it, right? Let alone machine learning and artificial intelligence and these things. So if you ever write a project for a PhD project or something, you have to put the magic words, CRISPR-Cas9, artificial intelligence, machine learning, because if not, you will be considered completely out of place, right? So that's how the science works and so on. Well, anyway, so I have to apologize that today, again, the lecture was a bit boring, let's face it, but I think that these are things that you have to learn and to study if you want to really get into this field, okay? But in any case, the only thing that I expect from this lecture is that you remember that there is a list of technologies that you can apply for genomic engineering, and that you have a choice of them, and that eventually, one way or the other, we have now all the tools that are required for starting with a given genome and entering all types of edits that, as I showed in the first slide, are related, at least conceptually, to the way an editor edits a text in a book or something like that. Okay, so now we have all the technologies to write and to edit text. Now the question is, what do we want to write, right? And this is another problem that is quite different and depends very much on the creativity of the people involved. Okay, so let's make a little break for coffee, and then we'll continue a little bit.